Well, most NMR is done by analyzing the proton in hydrogen atoms, but you can actually perform NMR on a variety of different atoms as long as the isotope in question has an odd number of protons plus neutrons. Then generally you can run NMR based on that element. And in organic chemistry, if we're not looking at the hydrogens and how they are connected in molecules, we're looking at the carbons. And so carbon-13 is an isotope on which we can do some NMR, and oftentimes it's done on the same instrument that does the hydrogen. And so as a complement to the proton NMR spectrum, you can run a carbon-13 NMR spectrum, and it tells you something about the numbers of carbons in atoms, just as we can count hydrogens by doing the examples we've been looking at. Now, carbon-13 is not very abundant. Only about 1% of all carbons have that mass number. Most carbons are carbon-12. But even with that small percentage, you can still get decent spectra. Because carbon-13 is rare, that means we don't have to deal with splitting patterns of carbon-13 interacting and being split by neighboring carbon-13s, because they're very unlikely to appear next to each other in a molecule. Carbon-13 does couple to hydrogen, but it creates such complicated splittings that it's hard to interpret the spectrum. And so as you see in this example for acetone, we're going to see that with carbon-13 spectra, all you see is thin spikes. We get nothing but singlets because these are said to be decoupled. The spectra are run under conditions to suppress the coupling so that the spectra are simpler to interpret. Notice the scale of chemical shift here. It still starts with a 0 on the far left, and we still use TMS as the reference, but it's the carbons in the tetramethylsilane that are given a chemical shift of 0. And notice the scale goes up above 200. So if you're looking at a spectrum, and even if it's not labeled as a hydrogen or carbon spectrum, you can tell which one you're looking at by just looking at the scale for the chemical shift. Hydrogen only goes up to about 12 or so, and again, this goes up above 200. So this tells us when we see more than one signal, it's how many carbons, or at least how many non-equivalent carbons you have in a molecule. And along with the hydrogen spectrum, this is another way to confirm a particular structure. Now, we saw the hydrogen NMR of acetone here a while back. Here's the corresponding carbon spectrum. The red carbon gives rise to this signal up above 200. Carbonyl carbons typically have very high chemical shifts, and methyl group carbons correspondingly a lot lower, this one around 30 or so, and both of these blue carbons would be equivalent. And so if we suspected we had acetone as our sample, running this carbon-13 would give us the predicted 2 peaks corresponding to the 2 different types of carbons we have. And just as with hydrogens, we've got a correlation table where we've got a range of chemical shifts that are associated with carbons in certain environments. We just saw in acetone the carbonyl carbon, and you can see the highest chemical shifts are associated with carbonyl compounds. Acids and esters give chemical shifts between 160 and 185. The highest values of chemical shift, you can see that's going to correspond to aldehydes and ketones. At the other end, when you have just CH3, CH2 group carbons in regular alkanes, they have the lowest of chemical shifts, just as those hydrogens have low chemical shifts in the hydrogen NMR. So again, this is of use sometimes when we're trying to verify a structure as being what it is. We can look for the presence or absence of certain signals in these different ranges. So we'll just kind of take a look at a few more examples of carbon-13 NMR. And again, in certain cases, that's going to help us to be able to confirm the identity of unknown compounds.